Good afternoon. My name is Uns Masad, and welcome to the second edition of the Spatial Omics seminar series. I am a PhD student in the Beaver Surf Lab at Yale University, and I will be presenting one of our latest technology developments in super-resolution fluorescence microscopy. But first, I'd like to motivate one of the reasons we need imaging tools that probe the nanoscale. In the context of the omics, bulk omics analysis that use average data across a bulk population of cells, although valuable, fails to reveal important biological phenomena. However, analysis of the genomes, transcriptomes, and proteomes of single cells in the context of their spatial information can reveal a wealth of information: tissue heterogeneity, spatial organization, gene expression and dynamics, and disease pathogenesis. And in the first edition of the seminar, we got a great overview on all the recent innovations made in single-cell spatial omics, as well as a glance at the promise of single molecule and super-resolution fluorescence microscopy in increasing the sensitivity of these methods. And now I want to ask, can we go beyond interrogating the transcriptomes of single cells to revealing the transcriptomes of and genomes and proteomes of single organelles or vesicle compartments? Can we push spatial omics technologies to the fundamental spatial scale, that of individual cellular compartments, or what we call cellular ultrastructure? And will that let us reveal previously invisible patterns and advance our understanding of biology in disease? These are all valid questions, in my opinion. However, we should first ask, do we have the appropriate tools to visualize all cellular compartments at once with nanoscale resolution in a cell that is extremely crowded? Can we look at cellular structures with the level of resolution and molecular information as is shown here in David Goodsell's artistic and fantastical interpretation of the Golgi apparatus? Can we achieve molecular imaging with molecular information? The answer, unfortunately, is no. That image I just showed remains art. The current imaging modalities that are used, however, to image cells at the nanoscale today are electron microscopy and fluorescence nanoscopy, or super-resolution fluorescence microscopy. EM, electron microscopy, has a spatial resolution on the order of nanometers. And it's been very successful at identifying membrane compartments in cells, such as the endoplasmic reticulum in this rat liver EM image taken by Yale's George Palladay. However, EM lacks throughput and molecular specificity, with immuno-EM being unreliable and limited to thin tissue sections. And it's also incompatible with transcriptomics and genomics. And although EM does achieve molecular resolution, it lacks molecular information. Fluorescence nanoscopy, on the other hand, has high molecular specificity. And this is by virtue of the contrast agents that are used, the fluorescent dye. Fluorescent dyes have high quantum yields, and they come in many different spectra, so one can do multiplexing and look at many molecules at once. And we can divide fluorescent nanoscopy into optical techniques and expansion microscopy, which is a sample preparation technique. Optical nanoscopy techniques circumvent the diffraction limit of light, which is around 300 nanometers, by switching fluorophores between on and off states. Examples include single molecule switching microscopy, or SMS, stimulated emission depletion microscopy, or STED, and 4-pi microscopy. And so with these techniques, we can achieve multiplex 3D whole cell images of at tens of nanometers of resolution, as I show here in this beautiful image of the Golgi apparatus from our lab. Expansion microscopy, on the other hand, is a sample preparation technique, and it is developed by the Boyden Lab in 2015, so it's pretty recent. In this technique, biological samples are embedded and hybridized to a superabsorbent hydrogel, and then physically enlarged or expanded by dialysis in water. And so if you have two labels that are closer than the optical diffraction limit of light, you can separate them in space. And then optically resolve them on a standard conventional fluorescence microscope. And so this makes expansion microscopy a super-resolution technique that bypasses the diffraction limit of light by decrowding biomolecules. And so in conclusion, with electron microscopy, we can achieve nanoscale resolution and ultrastructural imaging. However, we cannot routinely image cells in 3 dimensions, and molecular specificity remains a major hurdle. With fluorescence microscopy, on the other hand, we can image whole cells in 3 dimensions, and molecular specificity is a given, resolution is also very good and it's easy to use. However, we cannot look at the complete ultrastructural context of the cell, despite this improvement in resolution and contrast. And so this beautiful mosaic would look something like this with electron microscopy. We can tell that we're imaging the Golgi apparatus and clathrin-coated pits. However, we cannot identify the underlying biomolecules. With fluorescence microscopy, we get images like this routinely, right? So if we were to probe a protein on clathrin-coated pits, we can visualize them. However, we cannot achieve ultrastructural imaging. So the subcellular context of these proteins is lacking. And so what would it take to achieve imaging at molecular resolution and having molecular information? How do we create a tool that combines resolution, whole cell 3D imaging, specificity, ultrastructural context, and that is easy to use? What are the obstacles that we need to overcome this hurdle? If you look at this table here, you can notice that fluorescence microscopy actually satisfies 5 out of the 6 criteria we identified as the best tool to look at molecular, to look at molecules with information and structural context. However, fluorescence microscopy, as I mentioned earlier, does not achieve structural context imaging. And this is an issue. So what are the obstacles that exist to overcome this limitation? If we take a step back, we notice from this image that the cell interior is extremely crowded. Most proteins are on average 3 nanometers in size and approximately spaced 1 to 2 nanometers apart. And so how do we squeeze labels in? Even organic fluorescent dyes, the fundamental contrast agents, are also 1 nanometer in diameter. And so they are on the same order of size as the biomolecules themselves, making labeling of the whole proteome inefficient and susceptible to homofret interactions between the dyes and hydrophobic interactions as well. However, what if we separate individual biomolecules from each other by a large factor, something like 20-fold linearly, and then label the proteome in bulk? This way, the distance between proteins would be on the order of tens of nanometers, and bulk or pan labeling of the proteome would be efficient enough such that it would reveal local protein density variations and thus cellular ultrastructure. And so one would think of this concept as an optical microscopy analog to electron microscopy heavy metal staining. And so we set out to achieve this goal, capitalizing on expansion microscopy because of the biomolecule decrowding effect it has. Since its conception in 2015, expansion microscopy has seen further development, particularly by the Boyden, Chung, Von, Rizzoli, and Guichard labs. And we could divide expansion microscopy into 2 categories. The first uses proteases that chop up proteins to mechanically homogenize the sample, and this is to ensure isotropic 3D expansion. Whereas the second circumvents the use of proteases by reversing sample fixation instead. And so only denaturants that do not cut peptide bonds are used to homogenize the sample. The first approach can be done iteratively in a method called iterative expansion microscopy, or IXM, resulting in high expansion factors, so up to 20-fold linearly. However, protein content is lost, and the antibody labels must be introduced prior to expansion. And so the spatial resolution is still limited by the size of the antibody label. In the second approach, the protein content is retained. And so antibody labeling post-molecular decrowding is possible. However, it is limited to 4-fold because this technology is incompatible with iterative expansion. More importantly, all these techniques have been developed so far to achieve what current optical resolution techniques can achieve. That is to look at specific molecules with nanometer resolution. However, we are interested in cellular ultrastructure, the idea of seeing the whole as opposed to specific molecules only. And so we developed an expansion microscopy technique we call Pan-ExM, where the sample is expanded up to 20-fold linearly without the loss of protein content, and then labeled or pan-stained with an amine or thiol reactive dye that reacts with the bulk of proteins, for example, NHS ester. And we discovered that, in fact, ultrastructural context can be revealed with our method. The two major conceptual developments in Pan-ExM are the mechanism of the iterative expansion via polymer entanglement, and the second is what we call pan-staining of proteins, so labeling proteins in bulk. The first round of expansion in Pan-ExM resembles that used in magnified analysis of proteome map and ultrastructure expansion microscopy, which are variants of expansion microscopy that use sample denaturation instead of proteolysis to mechanically homogenize the sample. The only difference is that we covalently cross-link proteins to a polymer network here in blue that is synthesized with a cleavable cross-linker as opposed to a non-cleavable cross-linker. After this hybridization step, the sample is delipidated and denatured and then expanded by dialysis in water. And so note that these two figures are drawn approximately to scale. And so the proteins here do get separated from each other, as well as stretched and denatured. In Pan-ExM, proteins that are already expanded once, the blue polymers here, are separated from each other by embedding in another superabsorbent dense polymer network, this orange polymer, that will tangle with the protein polymer hybrids. This is because when the first expansion, the blue gel is cleaved with base hydrolysis, since the cross-linker is cleavable, the polymer chains become linearized and long. And so we hypothesized that they must tangle with the second cross-linked expansion hydrogel, the one in orange. To better illustrate this point, if we look at this protein here, if you look at this protein here, after cleavage of the first expansion hydrogel, the one in blue, we can see that it is still covalently attached to the now linearized first expansion gel. But it's also embedded in a second expansion hydrogel, the orange. The orange gel will retain the protein after it expands because of, again, the entanglements it forms with the blue linear polymer and protein hybrid. And the definition of entanglement in polymer physics is simple. If a chain crosses an arbitrary plane 3 times, then it is entangled. And this is illustrated here by Edwards' tube model. This type of double network gels, where one polymer is linear and the other is cross-linked, is called a semi-interpenetrated gel. And they have been used for decades to create mechanically stable gels and to entrap various molecules, proteins, and cells. Here we show that we can extend this mechanism, this concept, to expansion microscopy as well, to retain proteins and iteratively expand them from each other. Our second conceptual advance is what we define as pan-staining. It is essentially the labeling of proteins or subset of proteins in bulk. A first example is the labeling of primary amines on proteins with an amine reactive NHS ester dye. So this reaction, a second example is the labeling of thiol groups on proteins with malamide dyes. And a third example consists of labeling specific post-translational modifications on proteins, such as palmitate, after metabolic labeling with a clickable palmitic acid analog. And this is just a brief summary of the protocol, which I will not go through in detail. There are 10 steps, and it takes approximately 2 to 3 days to complete, from sample fixation to pan-staining. If we apply pan-staining to non-expanded cells, we essentially see no contrast or details. After a 4-fold expansion, we start to see the outline of mitochondria and the nucleolus, but really not more than that. However, after a 16-fold expansion, we can see the ultrastructural details of the cell. In this cell that is expanded 16-fold, if we zoom in into this region of its cytosol, we can distinguish at a first glance mitochondria, this mitochondria cristae here, and the stacking of the Golgi apparatus here. And it's important to note that this image was captured with a standard confocal microscope at a microscope facility at Yale. And its spatial resolution is around 15 to 20 nanometers, which is approximately the diffraction limit of light divided by the expansion factor. Inverting the color table, you can see that the level of detail in Pan-ExM starts to approach that of transmission electron microscopy. However, unlike electron microscopy, Pan-ExM is compatible with nucleic acid staining, as you can see here from this image. If you zoom in at this region and invert the color tables of both NHS ester and cytox screen channels, you can observe bright dots here that correspond to these bright circular shadows in the cytox screen channel, and you can see that they overlay perfectly. We believe that these bright structures might correspond to nuclear pore complexes. This is a different Z-plane of the same cell I showed earlier, and this region here is dark because the nucleus is out of focus. Again, these cells are expanded up to 20-fold linearly. If we zoom in here to this region of very dense NHS ester staining and invert the color tables of both the NHS ester channel and the cytox screen channel, we can resolve subcompartments of the nucleolus, such as the fibrillar center here and the dense fibrillar component. We also see an absence of chromatin staining at the region of the nucleolus here in the cytox screen channel. We can see that they overlay, in this overlay here, that chromatin wraps around the nucleolus. In some cells, we see even denser fibrillar components here, which correspond to denser nucleic acid staining, which we think is regions of newly synthesized pre-ribosomal RNA. With Pan-ExM, mitotic cells have distinct ultrastructural features, such as dense and smaller mitochondria here. And if we add a nucleic acid staining, we can see mitotic chromosomes here. And if we zoom in into these chromosomes, we can actually resolve their polymer brush-like structures. Pan-ExM is also compatible with vital stains, such as mitotracker orange, and this is a dye that covalently reacts with proteins on the mitochondria matrix in live cells. And we verify in this inset that NHS ester crystal-like patterns we routinely observe in our Pan-ExM images actually do correspond to mitochondria. Pan-ExM is compatible with immunofluorescence techniques. This is an IF against TOM20, which is a outer mitochondrial membrane protein. And so here you can see TOM20 in the context of the ultrastructure of the cell. Pan-ExM is also compatible with specific microtubule staining, opening the possibility of imaging the cytoskeleton in the context of a cellular ultrastructure. I show this here. In mitotic cells, for example, microtubule bundles are attached to chromosomes, here in red, via kinetochores, here very dark in this inverted image, NHS ester image, and very white here in this regular fluorescence image channel, NHS ester channel. If we zoom into these regions, we can resolve individual microtubules in microtubule bundles. So here you can identify 4 microtubules, here you can see 3, and this is the kinetochore. And this resembles EM images of the kinetochore microtubule complex one can image with electron microscopy. What about the Golgi apparatus? It's a major cellular organelle that tends to look ribbon-like in conventional optical microscopy. As I've shown earlier, it is possible to discern individual cisternae of the Golgi complex from the NHS ester channel alone. Here, to verify that this is actually the Golgi, we overexpressed a luminal medial Golgi marker, MAD2, and we found that it is perfectly superimposed with the stacks observed in the NHS ester channel. So if we zoom in here, we can clearly see individual cisternae of the Golgi apparatus, as well as multiple vesicular structures surrounding the Golgi network. We also combined this labeling approach with STED microscopy, which offers about a 5-fold improvement of resolution. And when combined with Pan-ExM, it should achieve a spatial resolution down to 4 or 5 nanometers, which is really on the order of individual biomolecules. In the NHS ester channel, which has an expansion-corrected scale bar here that is about as large as the diffraction limit of light, you can clearly resolve 5 Golgi cisterna. In the MAN2 channel, you can see 3 Golgi cisternae. MAN2 is a medial Golgi marker. This suggests that the other 2 cisternae here might be cis or trans. This combination of ultrastructural analysis with specific labeling at spatial resolutions down to 5 nanometers in this example could help map out specific compartments within organelles, as this example illustrates. And so we anticipate that with the appropriate biological questions, this approach could be very powerful. Unlike the Golgi and mitochondria, the ER cannot be inferred from the NHS ester channel alone. However, when we use immunofluorescence against an overexpressed transmembrane Golgi marker, such as Sec61 beta, this is a marker on this is a transmembrane ER marker, we can reveal the intricate and dense structure of the rough and smooth ER. When we zoom in here, we can clearly see hollow ER tubules and ER sheets that are fenestrated. To investigate structural preservation of the features I just showed at the sub-organelle scale in Pan-ExM, we quantified the distance between mitochondrial cristae, ER tubule diameter, and the distance between Golgi cisternae, using expansion-corrected values inferred from the expansion factor of nuclei. We found that the distance between mitochondrial cristae is about 85 nanometers, which is consistent with life cell STED data of HeLa cells. As for the ER, we determined an average tubule diameter of 47 nanometers, which is consistent with EM data. And as for the Golgi, we calculated an intercisternal distance of 64 nanometers, which is also consistent with electron microscopy data. With these results, we confirmed that sub-organelle structures are reasonably preserved with our protocol. To validate protein retention with Pan-ExM, we quantified ER and Golgi protein fluorescence signal levels after the first and second expansion. We discovered that actually after the first expansion, fluorescence signal increases, suggesting that epitope availability and molecular decrowding actually enhances fluorescence antibody labeling. After the second expansion, we see essentially no difference between it and the fluorescence signal in gels that are expanded once. And so this suggests that entanglement of protein and entanglement of proteins and linear polymer hybrids and cross-linked polymer networks is an appropriate mechanism for protein retention and hydrogel expansion. In ExM, biomolecules are separated from each other physically. And so it's important to validate that this separation occurs uniformly across length scales that are of interest to biologists. To validate the samples processed with Pan-ExM do not deform significantly, we evaluated the distortions of 3 biological structures: mitochondria, which I show here, microtubules, and the nucleus. To quantify sample distortions, we imaged the same structures, the same field of view, in this case, mitochondria before and after Pan-ExM. We then registered the post-ExM images to the pre-ExM images using a rigid body transform, which consists of uniform scaling, rotation, and translation. And then we used these rigid registered post-ExM images and registered them again to the pre-ExM image using a B-spline non-rigid registration. And here we plot the deformation vector field. We then calculate the difference between the vectors at any 2 points in the sample and across different length scales. This difference is the RMS error of expansion. The RMS error is about 2.5% for a measurement length of 10 micron for mitochondria, which is consistent with expansion errors reported in earlier detergent-based expansion microscopy methods. If we instead initially registered the post-ExM images to the pre-ExM images using an affine transform to account for hydrogel stretching. In Pan-ExM, this error drops to 1.5%. Similarly, for microtubules, the RMS error is around 2.5% for measurement lengths of 8 microns. If we initially use a rigid body transform, and this drops to 1.3% if we do an affine transform instead initially, again, to account for the stretching of the jaw. And finally, for nuclei, the RMS error is about 3% for a measurement length of about 20 micrometers. If we use an affine transform, this drops to 1.5%. And again, these RMS errors are consistent with earlier expansion microscopy methods, which report RMS values ranging from 1 to 5% for similar measurement lengths. And so this gives us the confidence that biological structures in Pan-ExM process samples are being separated with adequate uniformity. We then asked whether protein complexes are reasonably preserved with Pan-ExM. One such complex is the centrosome, which appears actually extremely bright in the NHS ester channel, since it's very dense in proteins. Fortunately, centrioles that compromise the centrosome appear perfectly circular in Pan-ExM process samples, suggesting an isotropic expansion at the nanoscale. And we infer this from a roundness measurement of 0.9. In fact, in U2OS cells, you can actually discern the cartwheel structure of the centriole. And this is strikingly similar to the first electron microscopy images of isolated Drosophila centrioles published in 1995 in Nature. If we invert the color table and take a closer look at these U2OS centrioles, we can actually see them in incredible detail. So for example, here in these dark regions, these likely correspond to microtubule triplets. We can also resolve the pericentriolar material, or PCM. In this top view image and in this lateral view image, we can discern the distal appendages, the subdistal appendages, and the satellite centriole structures emanating really from this mother centriole. And although it is not as resolved as this EM image of the centrosome over here, the improvement in resolution and labeling is significant. And unlike electron microscopy, this is compatible with antibody staining. For example, here we did an immunofluorescent experiment against poly-E, against polyglutamylated alpha-tubulin. And here you can clearly resolve 3 stripes, which should correspond to 3 of the microtubule triplets. And here I show an overlay. In Pan-ExM, imaging obviously is not limited to 2D imaging. 3D imaging is very straightforward. All you have to do is take Z-stacks. And this is an example. So these are mitochondria. Nuclear pores, the nucleolus, and the Golgi apparatus. The 3D data sets I just showed took 3 hours to record, which is relatively a long time. Again, these cells are very large. And so we teamed up with a group of researchers in UT Southwestern, specifically Rito Fiolka, Kevin Dean, Hannah Mariam McBeeb, and Bo Hui Chung, to image our Pan-ExM samples with their clear tissue axially swept light cheek microscope, or CT-ASLM. So what I'm going to show you is a 3D data set of a mitotic cell they imaged, which took 5 minutes to record. So here you see chromosomes and kinetochores. These very bright structures are mitochondria. And you can see a hint of the mitotic spindle. These are Philopodia. Okay. Okay. So yeah, again, this data set took 5 minutes to record. This is a 3D rendering of the same cell that I just showed you. Here in 2 colors, here in 1 color, NHS ester versus NHS ester plus cytox green here in pink. And with 3D data sets, there's a potential for image segmentation. These segmented cells, biomark clustered in our lab, show the nucleolus, nuclear pore complexes here in green, mitochondria in red, and Philopodia here in white. The next conceptual step we explored, and perhaps one that starts to probe the world of omics, is what we call differential pan-staining of the proteome. That is labeling specific subsets of the proteome, overlaying them, and then using the differential patterns in staining as another dimension of cellular state characterization. However, this goal would require that different subproteomes are in fact compartmentalized differentially, something the literature does not adequately explore due to limitations in staining densely crowded proteins, but something we believe makes rational sense, given that more and more studies today are showing that biomolecules on the nanoscale are inherently compartmentalized or organized and clustered depending on their chemical characteristics. And so examples of this include clusters of EGFR receptors on the plasma membrane, lipid rafts on the Golgi, etc. And so in other words, we hypothesize that the distribution of subproteomes on the nanoscale must be organized. To test this hypothesis, that the distribution of subproteomes on the nanoscale is compartmentalized, we combined NHS ester pan staining with metabolic labeling of palmitoylated proteins. Palmitylation is a lipid modification on proteins, and so we expect palmitoylated proteins to be localized to the membranous compartments of the cell. In non-expanded cells, palmitoylated proteins seem to occupy a significant volume in the cell here. However, we cannot tell what organelles this stain is labeling. And superimposing the palmitate channel with that of NHS ester does not reveal significant information. However, in cells that are expanded 16-fold, we can see how palmitate is localized to mitochondria, to the Golgi apparatus, and to ER tubules as well. In this more zoomed in overview image, you can also see how palmitate localizes to the nuclear envelope, the ER here, and mitochondria as well. If we zoom into mitochondria, we can see that palmitate nicely labels the membranes of cristae, as well as ER tubules nearby. And this is an overlay, and you can see this beautiful striped pattern. Yeah. Palmitate is also very enriched at the Golgi, where this post-translation modification actually occurs. And so you can see here how it's localized to the membranes of individual Golgi cisternae. Now that we confirmed that the palmitoylated proteome is spatially distributed across membranous subcellular compartments, we tested the hypothesis that the distribution of proteins with different amino acid compositions is also compartmentalized, extending the technique of differential pan staining to the fundamental building blocks of proteins, the amino acids. In the first example, we combined the NHS ester pan stain with a malamide pan stain that labels thiol groups on proteins. And we qualitatively looked at the distribution of proteins with different cysteine to lysine amino acid residues. If we pan stain the same expanded cell with both NHS ester and malamide dye, the resulting images look virtually indistinguishable. However, if we overlay them, we observe differential labeling in the cytosol. If we zoom in here, we see that Golgi cisternae show higher cysteine to lysine labeling ratio relative to the cytosol or to mitochondria, here observed by more intense magenta labeling. And this is another example where this differential pan staining of the Golgi is very apparent. So here, this is NHS ester, malamide, and then the overlay. You can see here Golgi cisterna highlighted with malamide staining. And we believe, in consultation with the Rothman lab at Yale University, that this is most likely because of the high levels of palmitoyl transferases in the Golgi, which proteomics studies actually show have very high cysteine residues. They're very rich in cysteine residues. And again, as for other cellular structures in the cytosol, such as mitochondria, the ratio of cysteines to lysines seems more or less consistent. In the cytosol here, however, you can see patches of lower cysteine to lysine labeling ratios, such as they're in green, but we cannot tell if these proteins are compartmentalized or if there's any organization to this differential pan staining. This is an open question. We believe that with higher expansion factors, we might discover even more differential pan staining patterns. But I hope in general, I convince you that through these examples, differential pan staining provides information well beyond the monochromatic electron microscopy-like contrast we can achieve with a single pan stain. This approach can be easily generalized to other subproteomes, such as the phosphorylated glycosylated proteomes, and we believe that this approach has great potential for automated segmentation of organelles based on differential labeling and perhaps ultrastructural omics. And this is something we are actively working on. And so now back to our question. Can we go beyond investigating the omics of single cells to revealing the transcriptomes, genomes, and proteomes of single organelles? What about the transcriptome? Is this compatible with expansion microscopy? So far, I've talked exclusively about protein retention in the context of expansion. However, we can use expansion microscopy for RNA imaging. By covalently anchoring RNA to a swellable polyelectrolyte expansion gel, one could retain RNA molecules in the hydrogel and then probe them after sample expansion. And so several groups, Boyden group and Zhao Zhuang, have actually demonstrated the combination of fish, here fluorescent in situ fluorescent in situ hybridization and RNA imaging. They call it XFISH, developed in 2016, as well as MERFISH and expansion microscopy, developed in 2018, and actually very recently fluorescent in situ sequencing and expansion microscopy developed again this past month. And so what about the proteome? This remains an open challenge. If we take this area of the Golgi apparatus here and we expand it with Pan-ExM, with STED microscopy or perhaps higher expansion factors, we should be able to see individual proteins, as the spatial resolution should approach 2 nanometers. And I've shown this, and I've explained this earlier with my Golgi images. So what if we now did the opposite of what we conventionally do with fluorescence microscopy. Instead of targeting specific molecules with prior knowledge of what they are, and if we use a top-down approach instead, where we start from the big picture here, in this case, the total protein content, and then probe different post-translational modifications or protein motifs, adding a different level of selection every time. With enough specifications, this could lead to the identity of the protein. This project is less, and that's something we like to dwell on, but we do not think it's impossible to achieve in the future. Yeah, so this project is less than a year old, and you can find more details in our recently published preprint. We are happy to collaborate or assist you with the protocol if you'd like to try it out. Please contact us anytime, and thank you for listening. With that, I'd like to thank all members of the Beaversdorf lab, and in particular, my PI, Jörg Beaversdorf, which is great, and his guidance was very helpful, Hannah Maria McVieve, Rito Fiolka, Kevin Dean, and Bo Hui Chung for the light sheet imaging, James Rothman and Andreas Ernst from the Rothman lab, Mark Lessard and Felicia Kidd from our lab.